being here, everyone. Today I'm going to be talking about some of what I worked on for the last 6 years, which is ecological diversification across different taxonomic scales in these 3 adaptive radiations of birds in Malagasy. Okay, so I had to start with kind of the same place I started my prelim presentation, which is looking at phylogeny. Phylogenies are really cool, I think. They're kind of a map of how we're all related and where we come from in the broadest sense. I think that's kind of the idea that first drew me to studying biodiversity. Oh, you're muted. I should no longer be muted. What first drew me to studying evolution is the biodiversity is just so incredible, and evolution gives us this opportunity to sort of understand where it all comes from. I also want to use this tree to illustrate one particular point that has really been a focus or a starting point of a lot of my research, which is this idea that this has really come to our attention as we have developed these modern phylogenies, that biodiversity is really unevenly distributed. So just to give an example of that, these over here are paleocnath birds. There's not a whole lot of them. They're sister to all the rest of living birds. And meanwhile, over here we have Passeriformes, which is a single order that makes up over half of all living bird species. Right. So it's a lot of different explanations we could give for that. One of the processes, though, that leads to that unevenness, kind of a focus of my PhD, is adaptive radiation. So this term broadly refers to groups like these Hawaiian honey creepers that have evolved an exceptional amount of diversity in response to some ecological opportunity. And so what we mean by that is, for example, in the case of these Hawaiian honey creepers, their ancestors arrived in the Hawaiian Islands millions of years ago. They're finches taxonomically, so they look something kind of like that. And then because there weren't a lot of other birds in the Hawaiian Islands, there's not a lot of competition. There's sort of these empty, we might say, ecological niches. They were able to radiate into all of these really, really different forms that do really, really different things in the environment. And so this interplay makes adaptive radiations these really great systems for studying lots of big evolutionary questions, but especially for thinking, I think, about how ecology shapes evolution. So I want to talk a little bit about some of those big questions that interest me most, that have kind of motivated this PhD. So first is, how can we bridge the pattern process divide? So what I mean by that is when we look at big groups of organisms, adaptive radiations, for example, like these cichlids that live in rift lakes in Africa, we observe these big picture patterns. They have an exceptional amount of diversity. They've speciated really rapidly, et cetera. And then what we often ultimately want to know is, how does this happen? And to really get at that, what we're ultimately trying to do is link these kind of microevolutionary population level processes to the broad macroevolutionary patterns that we observe. So I can't talk directly about everything that goes into this. It's, a huge, huge topic, but I'm going to go into a couple of the approaches that I've kind of drawn on. The first is to look at speciation. Speciation is kind of the link in the scale between population level microevolutionary processes and macroevolution at the level of clades. And to have ecologically diverse species, you need them to actually speciate. So some different ways this can potentially happen is we might have ecological speciation. In the case of these sticklebacks, which have been really well studied, they're able to diverge into these two morphs, these two populations, sort of incipient species, the pelagic and benthic ones, even though they're occurring in the same lake together, because the divergent selection for those different ecologies is strong enough to allow that to happen. We can also have sort of, at least not primarily, ecological speciation, where, for example, in the case of this cryptic species group of anthidas, they've evolved into all these different species, really just because they're geographically isolated from one another. And in terms of their ecology, they're probably very, very similar. They basically all look like that. But we can have also, of course, a continuum between these, including, for example, maybe non-ecological speciation. And then when they come back into contact with one another, they might subsequently diverge ecologically. And so in practice, these can often be really difficult to distinguish from one another. And to really like untangle the speciation process, you kind of need to know about these things, about ecology, about the genetics, and about the distributions of organisms. Another approach we could think about is ask what characteristics make evolving a whole lot of diversity easier or more likely to happen? What might make some groups of organisms more evolvable than others? There's, again, lots of factors that go into this. One I got interested in is essentially the capacity of traits to vary more or less independently of one another. So, right, organisms are really complex. They're made up of many parts. Some of those parts can be connected more or less closely to one another. This kind of organization exists at lots of different scales. But I think, for example, about like the bones in a skull versus the bones in a limb as an example of that. And potentially the relationship between this patterns of trait covariation and evolvability could work either way. It might be that if organisms are more modular, their sort of different parts are able to vary more independently, that might make a greater variety of trait combinations able to evolve more easily. And so, you might get more diversity more easily. But on the other hand, if different traits are really tightly integrated with one another in sort of functional ways, that might allow evolution to proceed faster, especially if it's sort of maintaining adaptive combinations in this way. And there's evidence to show that in different circumstances, this might work either way. Another big question that I've been interested in for a long time is how can we identify shared general principles? This started kind of when I was a kid, thanks to my dad being kind of a Gould groupie. Gould is sort of one of my introductions to evolution. And so, reading him as a kid, I got really fascinated really early by these ideas of like contingency versus predictability in evolution. Gould used the metaphor of replaying the tape of life. Would we see the same things? Would it happen the same way if we gave it another replay? And again, of course, there's tons of different ways to tackle this big question. Experimental evolution is one, but one that we really need to rely on at a more macroevolutionary scale is essentially comparing many different groups. Right, so we study many case studies essentially of how does evolution proceed, and then we compare them to one another and try to identify commonalities or differences and why those might occur. Okay, so I've talked about some of these big picture approaches to understanding biodiversity patterns, and I've talked a little bit about how adaptive radiations are really great systems for addressing a lot of these kinds of questions. And now I want to talk a little bit about the system that I worked in, starting with the setting, because I was, as Sushma mentioned, so incredibly lucky that I came here and got to work with adaptive radiations from one of the most incredible places on Earth, which is Madagascar. Skipped ahead. So Madagascar is pretty famous, I think, among biologists as this incredible hotspot of biodiversity, even if the movie didn't really do it justice. But there's a couple of sort of broad contributing factors to this. One is due to precipitation patterns, and it has a central mountain range. There's a ton of really diverse habitats. It's got humid rainforest, it's got this kind of dry forest, it's got this spiny subdesert, as well as that elevational variation. It's also been isolated for about 80 million years in pretty much its current location off the coast of Africa. And that means that the few groups that have managed to reach Madagascar, there's not a lot of other organisms there. And again, they had this amazing opportunity to diversify into tons of different endemic radiations. This is my chance to show you some non-birds, because they're also pretty cool. There's one bird. It's not one of the ones I studied. All right. So, this presence of so many different diverse endemic groups is something that makes Madagascar really special. And that includes the groups that I studied, which are its three main radiations of birds. So very briefly, these are the bangas, which are an endemic subfamily. They're not, but they're related to shrikes, so their relatives are rather shrike-like. if that means something to you. Tetracas, which are the endemic family Bernieridae, they're mostly kind of warbler-like. And kuas, which are an endemic subfamily of cuckoos. And the fact that we have these multiple radiations and that we have some baseline of prior work on these groups, some genetic information about them, but for the most part they are very little studied, makes this really great system for addressing a whole bunch of these different questions that I've been talking about in terms of diversification. Okay, so my sort of big goal of my PhD was to try and gain this more comprehensive understanding of some of these different processes and how they interplay with one another. And to do so, I ended up taking this approach of working at kind of different taxonomic scales. So I'm going to talk about my three main projects now. First, I looked at the level of a single species complex in order to study these processes of population divergence and speciation. Next, I'll talk about looking at diversification processes that happen at the level of a clade, including a little bit about what that can tell us potentially about evolvability. And then finally, I compared diversification across the three clades to get at that idea of predictability versus contingency in diversification. All right, so to start with, I'll talk about looking at the speciation process within a single species group, particularly these guys, Kua cristata. And the sort of two big general questions I thought about here were, first, is there species level diversity within this group? And second, how is genetic diversity distributed geographically in these populations? All right, so as I mentioned, to study the speciation process, we kind of want to know about the ecology, the genetics, the distributions of organisms. And phylogeography is really the field that unites these approaches. And there's a bunch of reasons we potentially might want to know about this. It's pretty important for conservation. We need to know what species exist and where they live if we want to protect them. This is showing a whole bunch of micro-endemic chameleons in Madagascar. It impacts our understanding of big macroevolutionary patterns. This is from Tyler Imfeld's work, a former PhD student here. And he found that when he added a whole bunch of additional cryptic species into his analyses, that changed some of his results of like the overall macroevolutionary patterns of the group. And then, as I discussed before, there's that speciation process itself. And this is something that my lab group has actually been working on in some different Madagascar birds for a while. So as an example, this dark Newtonia, which is a kind of vanga, has, it turns out, two very genetically divergent populations in the northern and southern parts of the mountains. They should probably be considered separate species, and they're separated by this lower elevation area that's a barrier to these montane birds. And this is, more consistent, at least with the sort of non-ecological speciation process. They're not in particularly different habitats. They just are genetically isolated. And then on the other hand, we have species like the Sufis vanga, where there's, again, very divergent groups, but these occur in the dry forests, the great humid forests of Madagascar, which suggests more of that process of ecological speciation, where they're adapting to these different biomes. So to keep on sort of growing our understanding of these diversification processes in Madagascar and in these radiations specifically, I chose to focus on the crested kua. For a few different reasons. There's been no detailed genetic work on any of the kua species before. And this is the most widespread one. It occurs in sort of all of Madagascar's major biomes. It has actually described subspecies that live in those different biomes, including the one in the southwest called Pyropyga, because it has these sort of orange undertail coverts. And it's sometimes been considered a separate species because it does look a little more different from the others. So in terms of the approaches I used, my main data set consisted of what are called ultra-conserved elements. This is a reduced representation genomic approach. And then we had sampling from across the range. I want to acknowledge my co-authors on this, in addition to Sushma Maheshwari and Steve Goodman, who were really key to, in addition to just helping to collect some of these samples, understanding these distribution, the ecology, what am I actually looking at here? with their decades of experience working in Madagascar. And then, yeah, so I use a few different approaches to construct phylogenetic trees that I won't particularly get into here, but we estimate these trees in order to understand, how are individuals, how are populations related to one another. And so I'm showing you just one of those trees here, and then I've mapped the individuals back to the locations where they were sampled. And then I also used some population genetic approaches to look more like how is genetic diversity distributed. So the particular plot I'm showing you here is the principal component analysis. Essentially, this is a dimensionality reduction technique. I have lots and lots of genetic variation. That's a very high dimensional data set. And this finds the sort of primary axes of variation within that. So this isn't all of the genetic variation, but it's, those first primary axes of it. And what both of these are showing you is we do have 3 pretty well-defined groups that do correspond to those previously described subspecies. So that's sort of unsurprising, makes sense. What was a little bit surprising is that the most divergent group, so the one that's was different on this first primary axis of variation, Cristata, that's the one that lives mostly in the far north and then a little bit down the east coast. And it's not the one that looks the most different, that had previously sometimes been considered a separate species. That southwestern one is more closely related to the other western subspecies instead. So the populations, are pretty well differentiated. They do align with these previously described subspecies. But we also found that even though they are quite genetically distinct, there's also some pretty clear signals of introgression. So there has been some hybridization between these different populations. So this is from an admixture analysis, basically, that identifies how many genetic groupings there are. We still found 3 that did align with those subspecies, but it also can assign mixed ancestry. So each of these columns is an individual, and you can see that the individuals from the locations where those subspecies sort of meet do have mixed ancestry to different degrees. So if we look at that on a map, each of the pie charts is showing those admixture proportions by each locality. And I'll highlight that in the West, where that more phenotypically different Pyropyga subspecies comes into contact with Diamanti, the other Western one. This boundary really is right where we see these 2 biomes coming into contact, the spiny forest and this dry deciduous forest. And you actually, if you look at photographs from that area, you can see sort of individuals that look intermediate. And so the introgression, is occurring here. It seems like this could be consistent with ecological divergence. It certainly, is aligning with those biome boundaries. Up north, though, the differences between habitats here are not really clear. Most of these northern as well as eastern Cristata actually live in dry forest that looks pretty much exactly like the dry forest that Diamanti lives in. And a possible hypothesis for what's going on here, although I would love to actually test this in some future work with better sampling, is that they might have formerly been separated by that area where the forest kind of cuts all the way across to the west coast, the Samburano. And potentially this could even be recently coming back into contact with forest clearance, producing more sort of connections for these edge occupying species. This was actually proposed by a guy back in the 1930s, and I would love to dig into it more because it'd be really cool if it was true. And then just another way of looking at this, in terms of this introgression, essentially what we're doing here, it's called triangle plots, awkward term, excuse me, but you assign alleles that are unique to each parental population, those are in the corners, and then you can calculate for this hybrid index. And essentially what we're seeing here is that these individuals are back crosses with some ancestry from one parent, but most of the ancestry from the other. And it gives you kind of a rough visual estimate of the amount of hybrid ancestry. So overall, we found that there's pretty substantial divergence, that divergence is aligned with phenotypic variation of those previously described subspecies, but also that there's introgression where those subspecies meet. So are these species? This is kind of an edge case, right? As so often turns out to be true. So one further way we tried to get at this is they used a software called BPP. Very simply, this is a Bayesian modeling approach that, if this means anything to you, basically looks at like the coalescent history of the different loci and it sort of looks at, is that more consistent with these being species or not? And you can use the parameter estimates from this to then calculate this genealogical divergence index, which sort of gives a rough heuristic of, do these look like species? So over here, the difference between those two Western ones is pretty low. This is sort of within the range of values that we'd usually consider not high enough to consider these separate species. So they're divergent, but probably not enough to consider them species. Whereas the divergence between cristata, that northern subspecies, and the other two is good, it's about twice as high. And that's in the range of, yeah, these might be species, essentially. So, we're never going to get a totally for 100% answer due to the nature of species. But it seems like there's at least, there certainly is some pretty substantial diversity within this group. And it's got some interesting geographic distribution. All right, so next I'm going to zoom out a bit and talk about diversification at the scale of a whole clade. And I studied this in Bengas, which are the biggest, most diverse, most impressive radiation of birds in Madagascar. And the big picture questions I was asking here are, one, what are their patterns of ecomorphological diversity in terms of like, how morphologically diverse are they? What are their patterns of diversity? What can that tell us? And then the tempo part in the title, how has morphological diversity accumulated over time? And this was published last year in Evolution. Okay, so Bengas are really impressively weird and different from one another. And due to convergent evolution, a lot of them appear a lot more similar to members of lots of different other bird families that occur around the world. Because they are convergent on all of these different ecologies. And so much so that a lot of them used to be considered members of separate families until phylogenetic work revealed that actually these are just one family, they're an incredible radiation. Citation. And so that there is, in fact, a single common ancestor of these birds that arrived in Madagascar and radiated into all of these different forms. And to actually say something, though, about sort of what are the patterns of diversification in these Malagasy Bengas, we need a comparison. And so in addition to studying the Malagasy Benga radiation, I also looked at their relatives, the rest of the family, the non-Malagasy Bengas, which are found in Africa and tropical Asia. So the different ecological niches in Bengas are not so much in terms of what they eat as how they forage, where they find food. So we know, again, from some prior research that the ancestral Benga, in addition to most of the non-Malagasy species, was a gleaner. Basically, that means they move through the habitat and pick food off of surfaces. From within that group, some Bengas, a single subclade of Bengas evolved the sallying behavior. Essentially, they fly out from a perch to pick up food. And then finally, from within the sallying clade of Bengas, another clade evolved probing behaviors, essentially sort of reaching into substrates to find food. These are really broad categories, as you can see from the pictures. Within each of them, there is a lot of diversity. So how we study this ecological diversity, at least in my case, is in terms of morphology. Morphology is often a pretty good proxy for ecology, and this is pretty clear when we look at Bengas. Especially in terms of their bill shapes as well as their foot shapes, which relate to sort of, how are they moving through their environment. So one great reason why morphology is really helpful is that it's often relatively straightforward to quantify. So I was able to do so using specimens in natural history collections, and I will talk more at the end about natural history collections. But it's really incredible to be able to use these old specimens, some of them 100 years old or so, in all kinds of new ways. And in my case, that was micro CT scanning. So I scanned specimens from a bunch of different museums with help from a bunch of people. And then I placed these landmark points on these digital models of their bones in order to basically quantify 3D shape in this more complex way. Also, oh, sorry, thanks to Ewan Lim, who's an undergrad who worked with me on this project. He really led a lot of the foot analyses and is a co-author on this paper. And then the other thing we need to study, right, not just trait diversity, but how it's actually evolved, is an updated phylogenetic tree. And this is, the tree specifically is work that Sushma did before I arrived, so I do not get credit for that part. All right, so I'm showing you another principal component analysis. Again, we're sort of reducing the complexity of shape variation to look at these primary axes. Here we're looking at about 70% of the total shape variation in this group, specifically in upper bill shape. And the sort of pretty clear takeaway here is this dashed line is encircling all of the non-Malagasy Vengas, and the solid line is encircling all of the Malagasy Vengas. So one, yeah, the Malagasy Vengas are way more diverse in shape than their relatives. But also, we have these really extreme outliers. In the case of bill shape, these are the helmet Vanga and the sickle-billed Vanga. So to try to understand a little bit more how Vangas were able to evolve so much morphological diversity, including those outliers, I want to bring back this idea of integration and modularity that I talked about in the introduction. Here, right, we're looking at these 4 anatomical modules. We have the cranium, the upper and lower bill, and the feet. And I use some different approaches to quantify how much shape co-varies within and between those regions. So, all right, these are partially squares plots. It's a similar idea to those PCAs where we're doing this sort of dimensionality reduction, but here we're looking at the primary axes of covariance between 2 regions. So the axes that maximize covariation. And some things we can observe here, there's not really some obvious differences between the clades overall, which I'm showing with circles versus triangles. What really stands out, again, is those same outliers. And what was really interesting to me, that stood out to me, is that not only are they, again, outliers in terms of these patterns of covariation, they're outliers in different ways. They're mostly showing up on different axes, right? Yes. So a challenge that this caused is that, across all the different analyses of integration and modularity that I did, the differences between the Malagasy and the non-Malagasy Vangas overall were really, really inconsistent. They basically depended on whether I included those 2 outliers or took them out, and all of my results would change. And I sort of did about 20 different versions of all of these analyses to try and figure out better what was going on. And when I put all the pieces together, I think it wasn't really clear necessarily that there were any systemic differences in integration and modularity between the groups. There wasn't an overall shift where integration or modularity was promoting diversification in the group overall. Instead, it was sort of an overall conserved pattern for the most part. But I also saw that generally conserved pattern could be broken by selection, I think, for these really divergent morphologies. And it could be potentially broken in different ways. And I guess the other lesson here is outliers are important and also hard to deal with statistically. So then when I looked at rates of trait evolution, the other part of my question here, all right, so on the left, I'm showing you a phylogeny. At the top, we have the non-Malagasy Vangas, and then we have the Malagasy Vangas with these colors showing the different foraging categories. And I used software called phase traits to sort of test different models of evolution. And then on the right, I'm showing you this disparity through time plot. Basically, the gray is showing our null expectation of how disparity, shape diversity would accumulate over time. And the black line is showing us what we actually observe in this group. And what we see from both of these, although it's hard for me to show it to you on the tree. You'll kind of have to take my word for it there. But we didn't see a shift in terms of the rate of trait diversification right when they arrived in Madagascar, like we might expect. They first experienced this ecological opportunity. Instead, we really see it start to pick up when these sort of derived foraging behaviors, the sallying and then probing groups, first evolved. And then it really, really spikes when we have the origin of specifically these two species. And so my sort of overall interpretation of this is that morphological diversification is really associated with this expansion of niche space from the evolution of those novel foraging strategies, and that can take a while. Maybe related to, breaking those patterns of integration and modularity, or maybe related to something else, like something that happened in the environment. All right. So finally, I'm going to take another step back and address these questions of kind of predictability versus contingency by comparing diversification patterns across all three radiations. Sorry. So I'm taking a similar approach here to essentially to what I did with the Vangas, where I compare each clade to its relatives in order to look at, how is it diversified relative to these groups that don't live in Madagascar. And then I ask, again, sort of when did this diversification take place, looking at that timing of diversification. So I previously talked about this question of how do we identify shared general principles. And one way that we do this is looking at what are sometimes termed natural experiments. So for example, the Caribbean anoles have been really extensively studied in this context because they're this example of essentially repeated radiation. Its ancestral anoles have arrived on these different Caribbean islands and diversified into a similar set of different ecomorphs, different, ecologically similar species on these different islands. And so that gets at this question of if you have, closely related species, sort of similar starting points, essentially, that, arrive on these different islands, do they then diversify in the same way or not? So you're essentially getting a sort of replication, almost like we're replaying a little bit of that tape of life again on each of these islands. So the existence of these three independent radiations of birds in Madagascar gives us a really rare opportunity to do a different kind of comparison that hasn't really been done very much at all before. There's some key features of the system that make them a really good group for comparisons. They're not particularly closely related to one another within birds. They have a shared distribution, right? They all diversified across Madagascar. They live in all these different biomes. And they're of a similar age, as best we can tell, which is pretty important because, they'd be a lot less comparable to one another if some had much longer to diversify, had experienced a, different history of environments because they'd been there for much longer than others, right? And so this lets us ask kind of the reverse question. If we have different groups start from different starting points in the same environment, how much do we see a similar, evolutionary response due to that shared, experience of the environment versus idiosyncratic individual responses because the groups themselves are different. They're sort of intrinsic traits. Yeah, so that. So as with the Vangas, I looked at this using morphological traits measured on museum specimens. Used slightly different traits. I had 2D morphological data that I got from photographs, and then also a set of linear measurements. These are faster to collect for a really large number of samples than CT scans are. And then again, phylogenies of each of these groups and their relatives. So we can, put all of this in that evolutionary context. Yeah, and I mentioned comparing them to their relatives, so I'm showing here just briefly some of those. We've got some Kua relatives and some Tetraka relatives here. Excuse me, I just want to mention that this was a huge collaborative effort. This was actually started by grad student, undergrads mostly, as well as some grad students of Sushma's back at Loyola many years ago, collecting a lot of this data. And when I came in, I had the opportunity to build on that, as well as sort of figure out the directions I was interested in taking it. So I kind of completed this data set, mostly filling in a lot of these sort of relative groups. Got to visit a bunch of museums over winter break. And yeah, I mean, this is huge. So I want to say this is a huge data set. There's a lot I would love to do with this. I think a lot of people would love to do with this. I think we really, oh God, my battery's low. I've only scratched the surface of what can ultimately be done with this. Maria, would you mind getting my charger from my backpack in my office?
Yes.
Thank you. I don't know why I forgot to plug my laptop in. But yeah, really only, I think, scratch the surface of what we would ultimately love to do with this data set that we've collected. But I'll talk about some of those results today. Yeah. So, as I said, just a few results. As a sort of a baseline to get us started, showing you PCA, this time of that combined data set of linear measurements of our Vangas again. And yeah, it's not totally different from what we found before. The Vangas are still way more morphologically diverse. There's sort of a similar, depending on the specific data set we look at, set of outliers and morphology. Because this is combined, we're seeing sort of our extreme forms in terms of bill shape as well as foot shape stand out. And yeah, so they're way more morphologically diverse. They evolved at much faster rates as well than their relatives. Kind of at the other end of the spectrum of what I observed were the Tetrakas and their relatives. So same linear measurement data set. Thank you.
Keep going, keep going. Yeah, get that.
We good? All right. So the Tetrakas themselves are in dark brown, and their relatives are in lighter brown. And basically, what we see here is they don't have more total diversity. They're a little bit less diverse overall, although most of the differences were not significant. But they do have much greater than expected disparity compared to those relatives because there's only like 11-ish species of Tetrakas and like 60 locustellid warblers, which are their relatives. And so they're essentially much more spread out in terms of their morphology compared to the locustellids. And if we look in particular at this region of sort of unique space, so there's only these Tetrakas, what we see here is these are species that live kind of in the mid-story or in the canopy of the forest, which I thought was really interesting because that is, in fact, the sort of way in which this group really differs from their relatives. Basically, all of the locustellids live on the ground or in the undergrowth. And clearly they do have diversity in some different ways. We can see that. But there's also tons of convergence where different genera within this group look really, really similar to one another. Cool. And when I did some, again, kind of macroevolutionary modeling, that is essentially what we observed. So I'm not going to explain a whole lot of this for those who don't understand this, I'm sorry. But essentially what we saw is that these Tetrakas, the Madagascar group, fit this model of Brownian motion where they're essentially diversifying at a constant rate through time. So morphological diversity was sort of continually accumulating at this sort of essentially constant rate. Whereas their relatives generally fit this OU Ornstein-Uhlenbeck model, which essentially is saying that their diversification was more constrained towards sort of an optimum or a limited set of possible morphologies, which essentially means that distantly related ones are less different from one another than you would expect, given how long it's been, how distant they are, which fits that idea of they have been constrained to this sort of more limited set of ecologies, presumably because most of them are living on these continents where there's tons of other species that are already occupying, for example, those forest canopy niches. And then very briefly, kuas were kind of a mix of these two general patterns. For the most part, they were more like the tetrakas in terms of having not more total diversity, but more disparity than expected. They also did have higher rates in some traits than their relatives. And then they also had this really big shape divergence in some traits, which in part seemed to be connected to, at least ancestrally, there being much more terrestrial, and so having kind of morphologies associated with that. So kind of overall, what we saw here is that there's evolutionary signals of adaptive radiation can look different, but I think we're sort of pointing to a similar broad process here, where in the Vangas we have this accelerated diversification, higher rates, way more total diversity, whereas in the tetrakas and for the most part in the kuas, we didn't see that same acceleration, but we did see less constraint. They're kind of more spread out. They were still seeing this signal essentially of ecological release. When they arrived in Madagascar, they had that lock of competitors. They had those sort of empty ecological niches and were able to diversify kind of more freely than their relatives on the mainland. I think kind of similarities and differences. And then, so when we looked at shared timing, we again saw some of what looked like some sort of shared signal in terms of timing. This is showing those disparities through time plots again, with the Madagascar Vangas and kuas on the left and their relatives on the right. And it seems like there's maybe this similar pattern of an initial increase around 12 to 10 million years ago, and then a peak kind of 8 to 5 million years ago. But the tetrakas don't show that, as best I can tell. So there's definitely more I want to do with this, but if this is, sort of a real signal, and if this is sort of actually congruent in time, it would suggest a sort of external role that would be causing this, shared timing, like climatic changes occurring around that time. We're sort of talking mid to late Miocene. There was a lot of climatic changes happening around that time. All right, so to summarize, in my PhD, I studied phylogeography of Kua cristata to better understand these processes of population divergence and speciation. Found some possible additional species, or at least pretty unique clades, but defining species is hard. And I found what looked like sort of a mix of these ecological and non-ecological divergence, speciation mechanisms going on. I then looked at morphological diversification in Vangidae to study diversification processes at the level of a clade. We found that morphological diversity took time to evolve. It didn't happen right when they arrived in Madagascar. There was this big increase in certain clades when these novel foraging behaviors first evolved, and also explored sort of this impact of extreme forms, because outliers are challenging, but also really important. And then finally, compared morphological diversification across the three clades of Vangas, tetrakas, and kuas. Found lots of different patterns of morphological diversity, different amounts, different types compared to their relatives. It did seem like there was some shared patterns in terms of evolving greater disparity and the signal of ecological release. And there seemed like there might be some shared timing as well, at least between kuas and Vangas. So spent 6 years working on these questions. It feels like I could spend a whole lot more on them. There's so much more to do. That's exciting. But I'll leave it here for today. So I want to start by acknowledging natural history collections. This is where I get to be sentimental and show you lots of pictures for a few minutes. But first, yeah,
I used specimens from 4 different institutions, Bell Museum right here, Field Museum in Chicago, the American Museum of Natural History in New York, as well as, although I didn't get to go myself, the Musée National d'Histoire Naturelle in Paris, as well as genetic samples that were from the University of Antananarivo in Madagascar. And I want to talk about, acknowledging those museums and those specimens, but also the sometimes complicated history of natural history collections. This is a collections acknowledgement that folks at the Bell Museum, especially other curators, wrote. Because collections do often have this really complicated history, in particular a legacy of colonialism. For example, a lot of the specimens that I use were collected on this Franco-Anglo-American expedition to Madagascar in the 1930s, and I think just that name kind of gives you a little bit of a hint of, some of why that was happening. And I'm really grateful for the museum community here that's worked, pushed to engage with that history. And I think, folks should continue to do better because, we try to work with a global community of researchers to develop, research and collection capacity around the world. So I'm showing you specimens that were collected, with collaborators in Madagascar whose counterparts are still in Madagascar in that second image. I also just want to, thank the people who work at museums who facilitated my museum visits and my specimen loans, as well as all of the CT scanning that I did of specimens, which was thanks to especially the scanner here being broken for over half the time I was working on this, was a huge collaborative effort across many institutions to get this done. Yes, there's a new scanner just in time for me to leave. I'm very happy for everyone else. Yeah, so, obviously it takes a huge number of people to do a PhD. Start with collaborators. My advisor, Shushma, you'll have another slide. And my entire committee, Sharon, Keith, Karen, and David were all just amazingly supportive and pushed me in all of the right ways and were there to meet with me whenever I needed it, even though it was sometimes just my anxiety talking, and I really appreciate that. All of my research collaborators, folks who, helped me collect data, helped me think, made all of this possible in so many ways. Undergrads who I helped me collect data and, got to, do projects that push this, this ultimate research forward. All of the undergrad and master's students and postdocs who formerly worked in the Ready Lab who helped collect all this data as well. Funding sources, of course, especially the Bell and EEB, that gave me grants to make this stuff possible. Okay, I'm gonna speed through these because there is time for questions, but Sushma, thank you so much for all of your support, all of your enthusiasm, all of the things you taught me. I tried to find some fun ones. Taking me to Madagascar, that was a pretty amazing experience to have. There's a photo missing. Whoops. Sorry. But everyone in the Bell Hertz group, I gave up on trying to list names on these slides, so I'm sorry, but you guys know who you are. All of the fun times, all of the silly times, all of the gross and messy times, preparing specimens. It's been amazing. You guys are the best friend group, the best support group. Made it a whole lot easier moving to Minnesota, not knowing anyone when I got here. I don't know why that one was later, but that's from Zoom times. The other, I think, most important thing I did in grad school was be a union organizer. There's a lot of wonderful things about academia, but a lot of things that could be better in academia and also just in the world in general. I've been so lucky in the opportunities I've had, and I really want to make that possible for more people. And this was the way that I was hopefully able to contribute to that. And I also just met so many amazing people doing this. Everyone in St. Paul campus, everyone who was ever in a committee meeting with me, my inaugural executive board buddies. All of you were amazing. Um, Thank you so much for teaching me and helping me grow as a person. Slide, advance. I think these are too large a file because I have too many pictures. All of my friends near and far. I think a lot of you are on Zoom right now. Some of you are here. Your friendship means everything to me, and hope we go on lots more adventures together. My family. So I'm really committed for this. I know much of them are on Zoom. We have a lot of fun together. And I can always lean on you when I need it, which I sure have this past month. So thank you for that. I tried to find these really long. And I'll take some questions. Thanks. Great. Yeah. Thank you for asking that. That was not a planted question, but I am appreciative. Yeah, so what I was sort of referring to, the northern or eastern subspecies cristata, it's mostly found in the north in this dry forest. It also extends really, really patchily down the east coast, mostly in these kind of secondary or edge habitats. It's not really in the rainforest, which is what we think of as the eastern biome. And so it is more widespread along the East Coast, but it is pretty limited, pretty patchy, and there just weren't a lot of samples from that East Coast. But it was interesting to see that that one individual, even though it's geographically really far from all the others, is completely nested right in the middle of all of the northern samples. And so that doesn't prove, but it also doesn't disprove that that could also potentially be kind of recent dispersal from habitat change, or just that those populations are really connected with one another. Yeah, Sarah. Yeah, yeah. Well, there's lots of different ways we can answer. It's really hard to come up with a species concept that works for everything. I think that approach we often use, at least for birds these days, is we really want a combination of evidence where we want to know about genetic divergence, but we also want to know kind of what are the actual dynamics going on there. You know, if there should at least be some degree of reproductive isolation, even if it's not complete, where they're not just totally freely interbreeding with one another. But yeah. Holly. Thank you. Yeah. Mm. Yeah. I mean, habitat destruction in Madagascar is massive. It's you know, been losing forests really, really rapidly. It's one of the poorest countries in the world, and so you know, there's a lot of habitat destruction as well as hunting and other things because you know people are trying to survive. Uh, and so, I mean, one thing is just how much longer are a lot of these amazing endemic species that have taken millions of years to evolve going to be there? There's lots of amazing conservation work happening, but it's hard. There's also absolutely an impact of climate change. We very much see that. Madagascar is more or less in the tropics, although it is kind of at the edge. Its the southern tip is actually over the Tropic of Capricorn. But we definitely do see an impact, in particular increased droughts from climate change, and so that also impacts things. I think it would be really cool to, and I think some people are, at least to a degree, tracking, you know, how our distribution's shifting. Is that bringing things into contact in different ways? That's one reason I think it would be really cool to look into this species in particular, in more detail and see like, are these actually recent shifts? Because that would be very cool to document if it's true. Pause. I think we need to- Yeah, there's a yeah. I mean, certainly in terms of researchers, tons of interest. I mean, Marie-Jean, who I mentioned, has looked at kind of exactly this, like some of these diversification processes in different species, in magpie robins and some other species, as well as, being a collaborator on these projects. As well as, lots of research that's more directly conservation oriented, because of course that's such a priority for so many people. And there are lots of, local orgs that are working to educate and also just expand financial opportunities for people in, a lot of places that are conservation priorities to try and, educate people, but also just make it, financially viable to conserve places, because that's often, of course, the biggest challenge. So I think quite a bit, and it was really cool meeting lots of different people working there, including like some of our field assistants who are amazing and so great about like teaching me the birds. I asked them like quiz me on the birds as we were, out in the forest, and they were great at that. So that was really fun. In some ways, I don't know. It's hard, right? But I think we need to wrap it up. But one more, okay, go for it, Dad. Hmm. Some of them a little bit. Certainly some of the endemic species. One thing that's really interesting actually is that the sort of new endemics, species that are endemic to Madagascar, but it's made that arrived, just kind of in the last couple of million years. It's just like a single species that's in the same genus as like other species that are, more widespread. For the most part, those are more habitat generalist and more tolerant of human habitat alteration. Not all of them, but a lot of them. Whereas most of the members of these older endemic radiations are much more specialist, much more dependent on, primary forest or other, endemic habitat types. So a couple of them can tolerate sort of secondary growth or sort of, matrices of forest and cropland, but for the most part, they're pretty specialist and really don't very well at all. Uh, Pigeons and host. Well, actually, the red foodie is an endemic species that is basically a host sparrow equivalent.